So let me know when I need to start.
Yeah, it's going on. So we are waiting for next 5 minutes.
Okay. So how can I get this onto the YouTube?
Go to the link with that. All right. Let's restart. Okay. All right. So we'll come to OGC 2020, and welcome to the OGC, especially for our speaker, Professor Dr. Naila Rabbani. Okay, how are you, Prof?
I'm very well, thank you very much. Norma, how are you?
Fine, thank you very much. All right. So, Prof. Naila Rabbani, okay, let's share screen. All right. All right. Okay, you can see okay the screen here. Okay, let's read okay the impressive okay background from Professor Dr. Naila Rabbani. Prof. Naila from University Qatar is a is a professor of the basic medical sciences, Age and diabetes research group, College of Medicine, Qatar University. At the same time, okay, she is ex-directors of the Zeeman Institute, Experimental System Biology, Warwick University, UK. All right. So today we're going to share some experience on the application of machine learning to develop diagnostic algorithm using metabolomic markers of protein, uh, glycation, oxidation, and nutrition in early stage decline in metabolic, vascular, and renal health. All right. So we want to share, okay, with Professor Dr. Naila how the powerful of machine learning, especially support vector machine, to diagnose some disease. Okay, this is the very impressive, okay, in research. Okay, Professor Dr. Naila Rabbani. You can see the H index in Scopus is very high, 55, and the citation increasing every year. All right. So when you look at the Google Scholar, the H index is very high, 62, with a citation 12,415. And we are very happy, okay, to share some knowledge, okay, on the title as we mentioned before about how the mathematical modeling of the machine learning called support vector machine, okay, to optimize the validation algorithm, all right, to discriminate based on the diagnostic okay, utilities. So without any delay, okay, we pass this session to Professor Dr. Naila, okay, to continue, and thank you very much. Okay, now floor is yours. The streaming is yours. So any question you can post through the talk chat at the live YouTube. All right. However, you need to register at the YouTube, okay, to post the question. Right. At the same time, I'm also post the link, okay, to check your okay to get the e-certificate automated after the presentation. All right. So let's we have some knowledge sharing. Okay, thank you very much.
Right, thank you very much, Norma. First of all, I thank you for inviting me to talk about or share my knowledge of systems biology, because I do employ multiple discipline to do my research, and that includes chemistry, biology, uh, clin- um, mathematical modeling, computer science. So I have employed all of these in my research. I would like to correct one thing, that I'm not the ex-director of Zeeman Center. Rather, I was a deputy director of the proteomics facility at the University of Warwick, and I was member of the systems biology team there. So As Noma said, that I'm going to talk about the application of machine learning to develop diagnostic algorithms using urinary markers of protein glycation, oxidation, and nitration for early stage decline in metabolic, vascular, and renal health. And I also will share with you the diagnostic algorithm for diagnosis of autism and early detection of osteoarthritis. Right. So the learning objectives for this presentation is, by the end of my presentation, I would like you to describe biomarkers of protein glycation, oxidation, and nitration, and their use of reporters of metabolic and physiological processes, and to describe the concept of health biomarkers and how they may be employed with machine learning for health screening. And then finally, to show how combination of biomarkers using machine learning is improving clinical diagnostic blood tests with application to early stage arthritis and autism. So why should we screen for early stage chronic non-communicable disease? And the reason is that these diseases are the major cause of premature death and loss of productive life in adults of westernized countries and in Southeast Asia. So examples of that is type 2 diabetes, cardiovascular disease, chronic kidney disease, arthritis, and autism. Screening could save lives. CVD is the major cause of premature mortality, detected and treated in the long preceding asymptomatic atherosclerosis. Sudden deaths may be avoided, and that's 50% of CVD mortality. Screening could improve effectiveness of treatment. For example, early stage intervention in arthritis may prevent joint destruction, pain, and disability. And autism, there is a possibility of remission if treated earlier. So screening saves money. It's also cost effective. Screening of prediabetes and implementation of diabetes prevention therapy and screening increases awareness of health impairment. Stage 3 CKD has only 9% disease awareness. If not detected early, the risk of premature mortality from CVD escalates fourfold. So that's why it is important that we screen early stage these chronic diseases. To do that, we need biomarkers. So biomarkers is important to screen for improved health screening and detection of early stage diseases. Currently, prediabetes is diagnosed at early stages using HbA1c, glycated hemoglobin, but the sensitivity of this is only 30 to 40%, and specificity is 90%. And for early stage renal disease, we measure EGF, estimate glomerular filtration rate. For stage 3 kidney disease, so although the sensitivity is 95% of this technique, but specificity is only 66%, and it is only applicable to people at the age of over 50. Cardiovascular disease, to detect early, we use coronary artery calcium measurement and carotid intima medial thickness, but for that you need specialized equipment, highly trained operators, and highly trained clinical experts to interpret the data, and it is very expensive. For osteoarthritis, normally it is detected early using arthroscopy by orthopedic surgeon, where inserting endoscope into the joint and check the morphology of the cartilage. And the other method is magnetic resonance imaging. Highly skilled radiographer is required, and the equipment is very expensive. And sensitivity of that is only 70% and specificity 90%. For rheumatoid arthritis, anti-CCP antibody test is used, but sensitivity is very poor, 61%. I mean, you might as well toss a coin to say if you have rheumatoid or not, instead of the anti-CCP antibodies. For autism, to date, only the way diagnosis is done by interviews and observations by expert consultant physician, but it is a lengthy process. It takes, and sensitivity is only 60 to 70%, and diagnosis can take 18 to 48 months. So we have been I chose to study protein damage. We have been measuring these biomarkers. So knowing that there is really not very robust biomarkers to detect early decline of health, I wanted to combine computer science to see if biomarkers I study can be used. And what I've been looking at, the damaged protein biomarkers. The reason for that is that the physiological protein damage is a continual structural and functional impairment of the proteome, and it occurs in vivo by oxidation to methionine cysteine residues and nitration to tyrosine residues, early glycation to lysine residues, and advanced glycation to arginine residues. So this is not a random process, rather this kind of reactive metabolite target amino acid at functionally important sites. So formation, clearance, and excretion of these that protein damage marker changes with health impairment, and these are related to health span and likely sensitive to nutritional intervention, because early detection can help you to intervene early, even by diet nutritional supplements or by diet control. And these biomarkers of protein damage are potentially rich source of biomarkers for health and chronic disease. But then the question was how to measure it. That's where the analytical chemistry come in handy. I started off as a medicinal chemist, but to measure, there was absolutely no robust way to measure these protein damage markers. So during my PhD, I developed this analytical technique using stable isotope dilution analysis, LCMSMS, to quantify robustly over 20 protein glycation, oxidation, and nitration damage markers concurrently, and amino acid metabolome for plasma and urine for free addicts. So common clinical analysis using this technology, LCMSMS, we can measure protein damage addict content of plasma protein, giving the measurement in millimole per mole amino acid modified. It gives you steady state level of plasma protein damage and reflects mainly damage in the vascular compartment. Then we can measure plasma glycation, oxidation, and nitration free addict in the plasma, and this gives you the steady state level of protein damage free addicts, reflects whole body flux of protein damage. There is some contribution from food for some addicts, and it is highly sensitive to change in glomerular filtration rate. We also can measure urinary glycation, oxidation, and nitration free addict, normalizing to creatinine, and again, it gives you the flux of whole body, flux of protein damage from whole body, and with contribution from some food for some ages only. So there is the family of glycation addicts. Here's the chemistry comes in. Fructosamine is really the glucose sugar modifies lysine residues, and this molecule, fructosylysine, is being measured for the reporting glycemic control. And then there are advanced glycation end products. So fructosylysine and other sugar can degrade to form very reactive dicarbonyls, and these then go on to react with amino acid to form these different type of markers. Pentosidine is a fluorophore cross-link between arginine and lysine, And then there is lysine-derived AGEs, carboxymethyl lysine, carboxyethyl lysine, and arginine-derived hydroimidazolone, which is the glyoxal, methyl glyoxal, and 3-deoxyglucosone are three reactive dicarbonyl, which degrades from fructose and lysine and from glucose. And they are between 5,000 to 20,000 fold more reactive than glucose. And when it damages to the protein at arginine and lysine residues, and these amino acids are normally present at the functionally important sites. And once damage occurs, the protein function is changed, and it can then damaged protein can degrade it. And then, of course, there is crosslinks. Glucospan is a major crosslink found in the cartilages of the in arthritis. And we also have a biomarker which reports only from a food dietary origin, because it is on from dietary origin. So we can really use this biomarker to report on the leaky guts. So the family of oxidation adducts are the cysteine oxidation markers, and then methionine sulfoxide, which is the methionine oxidized to form this marker, and lysine oxidation marker is L-lysine, arginine and proline oxidation marker is this glutamic semialdehyde, and these are protein carbonyls, and tyrosine oxidation can form dityrosine, and tryptophan oxidation can form formylkynurinin. And nitration addicts normally nitrate tyrosine residues to form 3-nitrotyrosine. There are other amino acids which are report on, for example, transglutaminase activity. This is a major crosslink, ECM crosslink, glutamyl lysine, and ornithinated protein. It's a terminal product of hydroimidazolone age degradation, and citrulinated protein is the arginine deaminase product in arthritis. Hydroxyproline is a marker of bone turnover. It also comes from skin collagen and food gelatin, but it's a minor source. And branch chain amino acids are linked to insulin resistance. So this is all the chemistry of these protein damage markers, which we studied them using LCM-SMS and in multiple conditions. And of course, with only these markers or one single marker, we were not able to diagnose any condition. So the question was how to combine these biomarker responses. So that's where we applied the computer science. We got together with the computer scientists and asked them that we would like to develop a diagnostic algorithm using the machine learning. Because as I mentioned, there is no single biomarker that can produce conclusive diagnosis. A combination biomarkers of features in a diagnostic algorithm is required. So algorithms are developed by machine learning, an application of computer science where the weighing of different features is optimized or trained on experimental clinical data. And then once the training is done, it is validated. The algorithm is then validated on independent clinical data sets. And algorithm development methods used there are multiple methods, and that is the list I used, but since then there have many more other programs can be used. So generalized linear model is one, GLM with elastic net is another, ensemble random forest, non-linear decision tree-based method, and sport vector machine uses hyperplanes and maximum margin. So now I understand there is X gradient boost and network analysis. Also, there is a program can be used. So to use these algorithms, we need to have access to relevant data and expertise to decide on variable selection. So that is very important. And advantage of this is that diagnostic using algorithm, this is a data-driven discovery process of logical stepwise analysis without preconception and bias. So here is the example of one of the machine learning programs, sport vector machine. This graph shows dimensional scatter plot for a two-class labeled dataset of outcome cases and control. The data can be separated by an infinite number of hyperplane, but here we only show three of them. And then hyperplane maximizes the margin between the two classes of cases and control. These are specified by the sport vectors here. So the maximum differences from data at this margin is give us the best algorithm. So we come up with the concept of health biomarker in our EU framework program 7 bioclaims. What it means is that the biomarkers of the transition from good health to early stage disease. So we wanted to find some health biomarkers where we really find out if a person is really uh, kind of moving into a good health to not so good health. So if you know that in advance, then intervention with dietary supplements or medication may then restore good health to those who will benefit before overt disease is established. So that we term as precision health. So really treating people what is required to be treated, not just stabbing in the dark what could we can use. So the precision health may improve public health by decreasing incidence of overt disease. So we hypothesized in our this bioclaim study that the urinary flux of protein damage by glycation, oxidation, nitration, and crosslinks may be health biomarkers. So we tested this by studying the changes in urinary fluxes in early decline in metabolic health, vascular health, and renal health, which may progress to diabetes, cardiovascular disease, and renal failure. So our study design was we recruited impaired metabolic health subjects. These people were with early stage decline in metabolic health, as judged by homeostatic model assessment of insulin resistant, HOMA-IR index greater than 2.5 milliunit per millimolar insulin and considered to be insulin resistance. And glycated hemoglobin, HbA1c, between 38 and 46 millimole per mole. So these are also considered to be pre-diabetes, and other clinical tests within normal range. So these subject, so impaired vascular health subject with carotid artery intermedial thickness was greater than 75th percentile for age and sex on the left and right side. Other clinical tests were within the normal range. For impaired renal health, subject with EGFR of 30 to 60 ml per minute per meter square, and serum cystatin C was greater than 1.04 milligram per liter. So this is the upper limit of normal range. So basically these people were healthy, but they were have a just touch of a condition of certain metabolic, vascular, and renal health was impaired slightly. And of course, healthy control. So subject we recruited with clinical tests within normal range, all of them. So we collected urine second void after overnight fast because we wanted to make sure that there is no biomarker coming from food in the urine present. So that's why we collected second void after overnight fast. So we prepared 3 kilodalton ultrafiltrate for analysis using LCMS, and we measure all the protein, glycation, oxidation, and nitration biomarkers I have shown you. We measure all of them in the urine. And then we put all of these, but this is the subject clinical characteristics, as you can see. This is the healthy people, recruited 55 of them, 44 impaired metabolic health, and 58 impaired vascular health, and 43 impaired renal health. And you can see all of everything is normal apart from just the HbA1c was significantly different, and the HOMA-IR was different from the healthy control. In this category, only the CIMT was significantly different compared to the control. And for renal impairment, only the plasma cystatin C and the EGFR was significantly different compared to all the other groups. So we developed two. This is the showing the subject data analysis plan. So we have total 200 individual recruited, and the first step was to develop two-class algorithm where we were comparing good health versus impaired health, and it has all of the people with three different categories. And this was the outcome. So we used sport vector machine algorithm that we tried all of these different programs, and SVM gave the best algorithm for these categories. So for metabolic health, healthy versus pre-diabetes, the feature which gave the best algorithm was age, BMI, and branched-chain amino acids. This gave the sensitivity 80%, specificity 87%, and the diagnostic odds ratio was 27, which is pretty good for very early detection, separating healthy versus the pre-diabetics. For vascular, again, healthy control versus asymptomatic CVD, the same feature gave the best algorithm with diagnostic or odds ratio 7. And for renal categories, healthy versus stage 3 to 4 chronic kidney disease, we found that the age, BMI, fructosylcysteine lysine, and valine, it is one of the branched-chain amino acid, gave the best feature, and sensitivity was 88%, specificity was 92%, and the diagnostic odds ratio is 79. So I would like to just mention that currently in clinical chemistry, there is methods or diagnostic methods are used up to for a diagnostic odds ratio of 4 and above. So we are here giving you the diagnostic odds ratio between 7 and 79. It is pretty good. It's really good. So the second so as I mentioned, it was two-stage diagnostic algorithm. The first stage was to looking good health versus the impaired. The second step, we wanted to separate that all these three metabolic impairment that really pinpoint the person suffer with metabolic health or cardiovascular health or renal health. So that would have given us to treat them with precision medicine. So again, using the sport vector machine program algorithm, we found that step 1 for healthy versus control, a healthy control versus all entries, we got the age, BMI, fructosylcysteine lysine, and valine. Sensitivity was 82%, specificity 77%, and diagnostic odds ratio 15. And then second step, we were able to separate these three conditions with diagnostic odds ratio 5, 4, and 10. So this is still very impressive when there is very slight difference. Excuse me. So why are branched-chain amino acids so evident in the diagnostic outcome? So reason for that is that branched-chain amino acid increased by increased production by gut microbiota, decrease expression of BCC catabolizing enzyme in adipose tissue, in inflammation, and ER stress, and decrease hepatic catabolism. So that is the reason why BCAA were picked up the best feature in the algorithm. So that's where it was increased. But in chronic kidney disease, decreased branching amino acids were decreased, and due to decreased interorgan exchange from impaired output by peripheral tissues linked to increased catabolism. So that is why branching amino acids so evident in the diagnostic outcome. So then we wanted to see how we can apply this diagnostic algorithm to assess decline in skeletal health. And for that, we studied osteoarthritis, rheumatoid arthritis, and other joint disease. So osteoarthritis is a progressive joint disease with joint inflammation and reparative bone response. It affects about 100 million people globally. It is the primary diagnosis in 2.9 million joint replacement every year. So rheumatoid arthritis is autoimmune inflammatory joint disease. Disease-modifying drug treatments are used. Early intervention would improve effectiveness. And other inflammatory joint disease, we call it non-RA, often self-resolving and difficult to distinguish from RA in the early stages. So why we chose to study this? Because there is no biochemical test for detection of early stage osteoarthritis, and tests for early stage rheumatoid arthritis, such as anticyclic citrullenated peptide antibodies, require refinement to improve clinical utility. For this, our study group, we recruited patients with early osteoarthritis, advanced osteoarthritis, early rheumatoid arthritis, advanced rheumatoid arthritis, non-rheumatoid arthritis, and healthy control. So we look at the collected plasma and synovial fluid from the patient, and from healthy control, we use plasma. So this is the heat map of damaged protein proteolysis product, free adduct, and protein residues in all of these categories in the plasma and in the synovial fluid. So we developed, using the algorithm, only the early OA sample from or protein damage markers from early stage of arthritis people to develop to our diagnostic predictive algorithm for who is going to go on to develop which kind of arthritis. So we again trained and validated a two-stage diagnostic algorithm for detection of impaired skeletal health. So first step was skeletal health versus arthritis of any type. And we have 181 individuals. We trained 52 sample. We trained algorithm using 52 sample and control and impaired skeletal health, and we tested using 129 data set independently for this, and outcome was area under the curve 99%. I mean, this is conclusively separating healthy from the impaired skeletal health, because a random outcome would have been 0.5. Then the second stage, we wanted to do the typing of arthritis. So which arthritis really a person is suffering. And again, area under the curve gave for EOA or early OA 0.98, conclusively that which person might be suffering osteoarthritis, and 0.91 for ERA. Remember, anti-CCP antibody only has 61% sensitivity. And for non-RA was 0.68. So using this outcome, Again, we found that SVM1 algorithm was the best algorithm, gave us the best diagnostic odd ratio. So step one, we got the 91% sensitivity, 92% specificity, and DOR was 116. So the features used here was that the free addict, plasma methionine sulfoxide, dityrosine, NFK, and formylkynurinin, 3-nitrotyrosine, CEL, CMA, hydroimidazolone, and pentosidine, and also hydroxyproline. So that was the feature used in our diagnostic algorithm. For typing, we have almost similar features, but this time anti-CCP antibody positivity was also used to get the typing of these arthritis. And again, as you can see, DOR for early OA is 103, rheumatoid 14, and non-RA 4. So using these blood test or diagnostic algorithm, we can conclusively separate very early on the person with impaired skeletal health and then typing them that which type of skeletal health impairment they are going to go on to suffer. So next, we wanted to look at the autism. So autism is another condition. It's a developmental disorder of children, mainly affecting social interaction, speech, repetitive and compulsive behavior, hyperactivity, anxiety, and difficulty to adapt to new environments with or without cognitive impairment. So what causes this? Then the suggested causative factors are genetic cause, but it is only evident in about 30 to 35% of the cases. So the rest, environmental factors, account for the remaining 65-70% of these children. So diagnosis is based on observations and interviews to assess behavior and communication characteristics. And the problem with this ASD diagnosis is that it can take years to get referral to expert and then further months and a year to diagnosis. And diagnostic accuracy is considered to be only 60 to 70%. So I wanted to look at the children with and without ASD. So we collaborated with University in Bologna and recruited children, about 38 children, cases of ASD, and controlled 31. And we collected urine and plasma for that. And plasma was then separated in plasma filtrate and plasma protein. So it was a comprehensive study where we wanted to look at protein damage marker in all of the compartment of the physiological fluid, especially plasma and urine, and see exactly what happens and develop the algorithm with twofold cross-validation. So we measure 20 amino acids, all of them, 15 damaged amino acids, and glycated amino acids are these, and oxidation markers and nitrated amino acid. Data was corrected for multiple analyte measurement, Bonferroni correction, to minimize false discovery rate. Then we perform machine learning using ensemble and sport vector machines with twofold cross-validation and repeated 10 times. So this is the outcome of our study. And for plasma protein, we've found that the typically developing control children versus ASD using these features, which is CML, CMA, 3-deoxyglucose-derived hydroimidazolone, and dityrosine, gave sensitivity 92%, specificity 84%, and the DOR 68%. And for plasma filtrate, CML and CMA, only two feature gave reasonable sensitivity and specificity with DOR 9, and in urine, GSA and pyrrolin gave 77 and 76 sensitivity and specificity with DOR 11. So this, using blood tests, we could diagnose with these biomarkers within a week, and at least we can identify who might have ASD, than to be waited for years to get the referral, and only based on qualitative measurements. So this is robustly quantitative measurement can give you the diagnostic of a very high diagnostic ratio. So this, as I said, diagnosis of ASD could be done with blood tests within 7 days. So potential use of a blood and urine-based biochemical diagnostic test, I have just shared with you. So what is now required? Further clinical validation of all the tests in validation, which are in progress. Health screen improves access to diagnosis of impaired insulin resistance and asymptomatic atherosclerosis and chronic kidney disease. Remedial lifestyle and therapeutic intervention may prevent progression to advanced disease. Early stage arthritis provides diagnosis of early stage arthritic disease to support prevention of progress to severe OA, and sports therapeutic intervention to DMARDs to cure RA. Blood and urine test for autism improves access and swift diagnosis of autism, supports early intervention, which may often produce remission. I mean, that is very important. Early intervention can normalize a person, or remission can happen, but once the disease is set in, that's where you cannot reverse a condition. So to summarize, biomarker selection is likely to be most beneficial when focused on mechanism of health decline or early stage disease process, because the proteins are the machinery of life, and damage to the protein is the closest to the phenotype of the disease. So all these biomarkers are really coming from the mechanism of health decline. So that's why we got this very good outcome of diagnostic algorithm. So conclusive diagnosis may be achieved by a combination of clinical variable and biomarkers in a diagnostic algorithm using machine learning and applications. First, a blood-based diagnostic test for detection and typing of early stage arthritic disease. That was the first we developed and published in 2015. Then we developed this simple blood and urine diagnostic test for autism, published in 2018, and the health biomarker screen for early stage decline in metabolic and renal and vascular health is published in 2019. So, of course, all this work as a biochemist, I couldn't do everything by myself. So you have to work together with other colleagues to bring colleagues together, chemists, biochemists, mathematician, and engineers, and the computer scientists, to take the research truly from bench to bedside. So that's what I do in system biology, and that's my research. So I hope you enjoyed the talk, and if there are any questions, I will be happy to answer. Thank you very much for listening. Over to you, Norma.
All right. Thank you, Prof. Uh, Dr. Naila Rabbani. Very interesting talk. All right. So there are a few questions here. Okay. The first is from Dr. Hafiza binti Harun. Okay. The question is the methotrexate adequate to control QA and RH? So this is a
Sorry,
question from
can you repeat
Hafiza.
the can you repeat the question? Can I see it?
Is methotrexate adequate to
Okay,
control
very
QA
good
and
question,
RH?
Hafiza. I have been involved in a clinical trial where they were giving methotrexate to reverse early OA in the knee. But unfortunately, it didn't work. So they still haven't published the data. So it is not really good enough. But because, low dose did not really help the OA, but there are many other uh, things, dietary intervention would be more appropriate than this.
All right. The same question comes from Hafiza binti Harun. Right. The question is, as a result of your research, is there any natural medication, such as
Yeah.
food, supplements, or exercise, to reduce further aggravation of AQA, OA, and
Yes, we
RH?
are actually working on the food supplements, and based on scientific mechanistic understanding, that's where maybe some other time we found out that one of the transcription factor, NRF2, oscillate, and we have mathematician making this a whole programming it and mathematical modeling, and that NRF2 really induces all the protective enzyme which we need to be healthy. So we found that two dietary bioactive can activate this NRF2 and can prevent inflammation. And those two dietary bioactives are resveratrol and hesperidin, and they come from resveratrol from grapes, and hesperidin is extracted from the oranges, but they only work together, not individually. So when you take in synergism, they work synergistically. So we use these two, combination of these two, and we uh, reversed insulin resistant in overweight, healthy, obese people. And during this clinical trial, we found that it decreases inflammation, all the markers which are involved in osteoarthritis or skeletal health. So we are now, inshallah, working with the clinician to test that, and hopefully that we will have this capsule produced and bring to the market soon, inshallah. So we are working on it. But otherwise, there are other dietary supplement people say might help, but there is no clinical uh, proof for those. But the one which we are working on, we have clinical data, and would be available soon.
All right. Okay, the next question from Dr. Afiza. Okay, let's finish the question from Dr. Afiza. I guess the studies on diagnosis. Okay, are there any studies to stop further aggregation that you know of?
That is, I mean, previously all pharmaceutical companies have been developing these drugs to treat a disease. But now everybody knows that treating disease is not helping, because once the disease have established, reversing is not possible, and all these drugs come with the side effects. So you take for one condition and you end up developing many more. So now there is a World Health Organization and European Union. They are pushing for early detection and prevention strategy. And that's where we have these developing these diagnostic algorithms, because when you detect early, you can reverse early, prevent it. And we have done it with our clinical trial. We reverse insulin resistant to totally normal with these dietary bioactive without any side effects. So to stop these, again, these same bioactive or these dietary supplements can delay the diabetic complication. That's another strategy we are pursuing, that people who might go on to develop diabetic complication, retinal disease, kidney disease, and nerve disease. So taking these supplement can reduce the risk or delay the development of it, so keep you healthy longer. But uh, you know, once the disease is established, that in skeletal health situation, you have to, as a sport scientist, if I understand correctly, you have to keep moving. Your joints have to keep moving, exercising, and it helps.
All right. Okay. This is now it's come from my question. All right. Okay. Now you have so many data. All right. So there's so many data. I'm really impressed. Okay. You can access to the relevant data. Okay. Let's share with me how easiest to get a data,
Well, I
especially to
would be
diagnose.
Yeah, so I understand, Norma, the frustration of computer scientists and mathematical modeler is that they never get a robust, good quality data, because the data it is very important that the data is reproducible, it's good quality, and uniform. So what I have produced using this, as you wanted to know the integration of the science, and that's here it is. I, you know, to get a good science, to do a good data, you have to develop, you have to have robust techniques. So the LCM SMS technology is the best technology when you use internal standard, and you can get exactly what you find in physiological system, and it is reproducible. So I've been using this LCM SMS over 20 years, and I have data of from every condition, you name it. So currently I have over 300 data set from patient on arthritis where we are validating this diagnostic algorithm. So yes, I will be happy to share it with you, but unfortunately, um, there is even HbA1c, there is no standardized data. You will have from one clinic different set of standards and the others different. So that is the problem. And that's why we didn't have a very successful diagnostic algorithms, because there are problems that have solved that. So I can tell you with confidence that the data I have collected is robust and quantitatively very good, and that's why we have been successful in developing these algorithms.
All right. Nowadays, okay, people looking for the big data, right? So how you categorize this one is big data or small data that you're using to apply machine learning, support vector machine. Okay, you're doing some feature extraction, right? And then you
Mm.
are also using some software to support the process of simulation. So, do you think about the big data, right? Now, for example,
So yeah, yes, the big data, you could see, I mean, all these Google and these algorithms, they are using everybody's demographic of people, have developing algorithm, how to personalize advertisement to people. And that comes from the whatever they have for uh, our characteristics, what we all put it online. So that is the real data. And that's how they are so successful. But when it comes to the clinic, the big data is still patchy. There is this genomic, big studies, thousands of patients, and they're measuring genomics, and then they could develop algorithm. But genomics can only be good, as I said, genes are only report 30 to 35% of the condition of people. The rest are environmental. So that's why you have to come down and look at the not just genomic, transcriptomics, proteomics, and metabolomics. And that's the big data. But in clinical settings, still there is a problem to have a robust data. So big data, if you are social scientists, you can get from all these, people's data. That is okay. But on clinical setting, it's still patchy. There are uh, scientists that aspire to develop big data, but we are not there yet on this.
all right. So
So
now
what I
let's we stop about data. So there is a question from here, okay, Prof. Tahir. The question is: some people claim that diabetes can be reserved. It is true. If yes, how?
It can be reversed, but if treated early on. So when you are, let's say, pre-diabetic, and you can change your lifestyle, you can exercise, and it can be reversed. But even at the supplement we are developing, that we believe that diabetics, when diabetes is developed, your beta cell are still functioning. It's as the time passes and low control, that's where the damage occurs. So within 3 years of diabetes, with lifestyle and exercise, it can be reversed. Healthy eating. And it is true. It's afterwards, once you develop the disease, that's where, and it's established your beta cells are damaged, then it becomes difficult to reverse. Then you only can manage it. So yes, it is true, it can be reversed.
All right. So, wow. Now we receive a question from Arun. Okay, is the ML algorithm, okay, machine learning algorithm, available open source in any hosting service like GitHub or like machine learning or artificial intelligence? Means that, okay, the question is can we do use the diagnosis algorithm based on the machine learning at the open source environment or licensing, okay, licensing software.
Yeah,
Okay, in this case, they said that GitHub, okay, like ML and AI. AI is artificial intelligence.
yeah. So developing for, I mean, let's say if I want to transfer or translate my clinical diagnostic algorithm to clinical chemistry, for that we will have to produce a software for, and that can be then licensed to clinical chemistry lab. So that is, and because I'm not a computer scientist, maybe Noma can help how to develop a software. I have colleagues in computer science who are willing to do that. So that is the idea. We will have a software specifically where you can put the features which I mentioned in there, and after, of course, blood test and measuring that, and it will give you the probability of the person is have a condition or it doesn't. So yes, software is required, and that Noma, any suggestion on that?
Based on our discussion, okay, I noticed that you are using a few software. All right, you asked me some software which is not in my mind, all right, because this is a specific software for the specific data simulation, data analytic. So I'm not sure this is an open source or licensing. In
Or
Malaysia,
it,
we um,
yeah. So it won't be an open source. The it would be a licensed, I think. Well, I'm sorry, I'm not expert on this, so maybe
I'm not sure
any.
you expert or not, but okay, my friend here, okay, Prof. Tahir said that have you published your work in any mathematical journals?
Not in mathematical journal, but this oscillation we have done that have a huge mathematical component to it, that was published in Redox Biology. Oscillation, I can send you the link, Noma, you can share it. But we have not published in a mathematical because it has a very much biochemistry or clinical component to it. So it was published in a scientific journal of biological journal.
All right. This is your okay. Okay. This is okay. You can see here. I'm sharing the some journals that you are editorial board. For example, scientific reports, associate editor for BMC, endocrine disorders. All right. So
Endocrinology.
yes, all right. So now the next question, all right. Since I want to share, I'm stop sharing. Okay, now I want to share some question, right, about the all right here. Okay, okay. My question here. Okay, I saw this is my question. Okay, you okay expressed here there is two steps of diagnosis, okay, health control and early stage. So can
Mm.
you explain to us how to get a data based on the two stages here?
So what we have, so we recruited 200 subjects. If you go back to the other previous slide, there we specifically recruited people who have impaired metabolic health, impaired vascular health, and impaired renal health, and also healthy control. So these people were very well characterized. They have all clinical data on them, and only as inclusion criteria, only few relevant clinical characteristics was different from the normal. So it was not huge difference, very small. So this was in our European biofilm study, Austrian colleagues, they recruited them, and then we have the urine to measure these protein damage markers, if those. So that's how we have the data. And what we wanted to first, we wanted to see, because it was impairment of health was very early stage. I mean, at that stage, there is no test. It's just a person tells you, I'm not feeling very well or that. And then only if they're lucky, they go to have a test done of this, like, I mentioned. So we wanted to see if we can separate healthy from the impaired first. So that was the first step. So we have all 200 individuals used their data and trained and validated the algorithm just for that is two class. And then we only use the impaired health people, which is about 145 of them. And we developed three class algorithm where we wanted to separate each class with the different features. So that's how we developed this first and second step diagnosis.
Oh, okay. Right. So since very clear, okay, the way how you categorize the data, and I'm really impressed, okay, how synergize, synchronize between the subject, data analytic plan, and then the activities in the clinic, okay, for the ASD or for diagnose the autism. So how you compare, how you match, okay, between the two parties, all right?
Yes. This is a tricky one because these are children. So for many studies like ASD, so normally they recruit their siblings if they have almost similar. So it was age sex matched uh, um, uh, recruitment. So most of the ASD children have their siblings. So those were then recruited as control who didn't have ASD. So that's how they recruited children, and the parents were involved, and to make sure that they are perfect match, cases and control are match.
All right. Okay, so thank you very much, Prof. Okay, I think it's over. Okay, the questions is so many. All right, let's we stop the session. Right before we stop the session, okay, let's stop sharing. I want to share the attendance for today. All right, let's me share. Okay, you can go for the scan the QR code. All right, and then you will get the e-certificate automated after this program. Okay, let's okay, scan, or you can go to the link. Okay, this is the link. Okay, attendance for today. Okay, you can write. Okay, and or you can click to get the attendance. Okay, the next is, okay, we really appreciate, okay, want to, okay, share screen, okay, the certificate prepared by the university. All right, so Prof, we are going to send to you, okay, the certificate. All right, we hope that okay, so if any problem for the okay, the collaboration or affiliation, let me know, right? So thank you very much. I really appreciate, okay, all the talk today, very impressive. We can do the integration between, okay, bulges and machine learning, okay, how machine learning do a very good predictive method, okay, for your data set. All right, so thank you very much. Okay, without any ado, okay, we stop this session. Okay, there is appreciation from some friend here, right? For example, okay, Prof. Tahir. Okay, Prof. Tahir also there. They notice your publication at the scientific reports. Wow, really impressed. All right, that okay, for other session, okay, we hope that you can answer okay this question. Just scan the QR code and you can notice that we are now diagnose some disease which is dangerous to our body, especially for internal organ. All right. So thank you very much. Okay. See you for the next session. We have other session. Okay. Let's we share with you. Okay. For tomorrow and this evening. Okay. Okay. All right, here okay, we have a session and then we can scan the QR code and you can get to the website and scan the link. Okay, so thank you very much. Okay, without any delay, okay, we stop this session. Okay, Prof.
Thank you. Thank you
Thank
very
you.
much, Noma, and thank
We,
you, Dr. Tahir.
thank you.
And
Thank you very much. Okay,
okay,
see
then
you.
bye for now.
All right. Bye bye.
Bye.
All right.